All right. Good. Okay, there we go. And so here in the Steve Frank lab, which I'm a member of, they've been recently examining some of the tree responses to urban stress. And so I'm just going to give this as background to this citizen science project, which I'll talk about in just a moment. So they've studied willow oaks in Raleigh. It's a pretty common street tree. Willow oaks do have this important pest, the oak licanium scale. It's various species, and so previous graduates doing the lab looked at willow oaks across Raleigh, including licanium scale, and to see what happened. And so there's some figures here, but basically the areas in blue were colder trees, and the bars in red were warmer trees. And what this previous student, Emily Meineke, found was that at all these different stages of this pest, the licanium scale, they were about 8 times more abundant on hot trees than they were on cool trees within the same city of Raleigh. And so we had pest increases with urbanization increases. She also found that the trees that were hotter in Raleigh, they sequestered an estimated 12% less carbon than trees that were just 2 degrees Celsius cooler, again showing these impacts of urbanization and warming on tree growth and the environmental services it can provide. And the Frank Lab also works with red maples, which are also a very common tree. And so this was with another graduate student from the lab, Adam Dale. And so gloomy scale is a very important pest of red maples here in Raleigh and across the southeast. And as far back as 1912, it's been described as a very important and perhaps the most important pest for red maples. And so it's another pest we have here in Raleigh. And indeed, the amount of scales increases as temperature increases. And this figure is showing that with just a 2 degrees Celsius increase in temperature, it increases the abundance of gloomy scales by 200%. And so I'm going to show a slightly more complicated figure, but what this figure is showing basically is on the horizontal axis is scale abundance, and we have a probability of tree condition on the vertical axis. But as scales increase, your probability of being in poor condition increases, and the likelihood that you're going to be in excellent or good condition decreases. So another way to think about that is just to look at the number of red maples from hot and cool sites and whether or not they're in poor or excellent condition. And so here we're thinking about trees in hot sites, and we look at the ratio here. You're more likely to be in a poor condition tree if you're on a hot site in Raleigh. This is for red maples. And then if you're a cold tree, you're much more likely to be excellent if you're at a cold site. And so here, what we've shown so far is that urban warming and urbanization can increase scale insect abundance, and warming can also increase scale insect abundance, all of this reducing the tree condition. And so a less than 2 degrees Celsius increase in temperature can reduce the tree condition so that the hottest sites have over twice as many trees in poor condition than cooler sites, which represents conditions that may become more common as urban and global warming progresses. All right. So the question we're hoping to answer with the Citizen Science Project is, how do trees respond to environmental stress and pests? And that's going to hopefully allow us to predict the effects of climate change and urbanization on carbon sequestration, but also on tree condition, and trying to understand what plants we can plant in the right places. And so we sort of have an answer, at least within Raleigh in these urban areas, and we found that these warmer trees were in poor condition, had greater pest abundances, and also decreased carbon sequestration. But again, this is just for one city and one sort of habitat type, urban. So this is just trees in Raleigh. And so how do trees respond to environmental stress and pests across large areas? Because currently it's unknown how trees in different habitats and across latitudes will respond to climate warming. And so here these images are just showing this gradient between rural and urban. And we might imagine that trees along this gradient might experience different conditions. They might respond differently, as well as latitude. While urban warming may affect trees by increasing these stresses in areas that are cooler, warmer, trees at higher latitudes where things are cooler might actually benefit from warming as growing seasons could increase. But ultimately, this information across this large geographic range and these ranges of urbanization are necessary to select suitable planting sites for planted trees. And so we need information across these large geographic ranges, but that's going to require hundreds of thousands of measurements, which is a lot. And so I talked about some of the efforts we've previously done in the Frank Lab, and we had relatively few trees to that, and we did pretty intense measurements for each tree. But in order to really get this good idea across this larger range, we'll need all these measurements, and this seems like a great opportunity for citizen engagement and for teaching and learning. So teaching about the importance of trees and the benefits they provide, but then also allowing us to learn by getting us additional data sets that citizens help collect. And so now I'll talk about this project, A Tree's Life. So when we initially launched A Tree's Life, we're going to be working specifically with red maple, and we're going to do that because it's one of the most commonly, if not the most commonly planted street tree in the eastern and specifically the southeastern U.S., which we're hoping is going to allow us to have a large potential pool of participants. And also giving this large range of latitude as well as urbanization gradients. And so to do this, we hope to recruit master gardener, master naturalists, and other volunteers. And right now we're planning to do this through our collaborators and cooperators in both Extension and other university departments. And the project does have a website. It's a little obscured here, but it will be again throughout the at the end of the presentation. And so I'm going to talk just a little bit about what we're asking folks to do with this project. And so if you think you have someone who's interested or you might be interested, what do you need to participate? You need a red maple in your yard. That's all you really need right now. There's a few specifics, and we're going to go over those real quick. And so that red maple needs to be approximately 6 to 11 inches in diameter. Go over how to estimate that if you don't know already. But here, this is the form that links from our website, and this is how you would sign up. It just asks for some basic information, including your name and your address, if you have a tree, etc. And so it's going to show you a picture like this, where you're just showing you how to estimate your tree's diameter with the ruler. And so what are we doing with these folks with these trees? Well, what are we hoping to collect? Well, we really want to monitor tree trunk growth, specifically the diameter at breast height. This is just a standardized height, 54 inches, for which people measure the size of the tree trunk. That's so the diameter at breast height. And so what's going to happen if if they have a tree and they sign up and they want to participate? Well, we're going to send them a kit. And effectively, the kit is just going to be this dendrometer here, which is a plastic band with a spring. And it's also going to come with instructions, of course. But effectively, pardon me, jump back quite a few slides here. So effectively, most of the work that's involved in this is signing up, because we're going to mail you this band. It's relatively easy to install on your tree. It causes no damage to your tree, and hopefully we're just asking folks that can leave it on the tree for at least a year, if not longer. And as the tree grows, the spring expands, and so does the band, and it just marks how big your tree is at that breast height. And so some examples of the instructions. We have instructions of where to place the band depending on the certain nuances of your tree's growth, whether it's growing on a slope or on an angle. Again, a pretty simple installation procedure, but that doesn't mean the data is not important. It's just going to give us some really important data in order to be able to, again, evaluate how trees respond to urbanization and warming across a large geographic range. And so, that's the wrong one. So after installation, we're just going to ask folks to upload a photo, and that's just going to include a picture of their dendrometer, and occasionally enter readings. And this is all going to be web-based. And so if you can take a picture with your phone, there'll be an option there to send it to us, and then also you can measure the tree's growth periodically. In addition to the dendrometer band, your kit is going to come with this wonderful manila envelope, and it's a pretty advanced scientific piece of equipment, but we're going to ask you to hang that on a tree with a paper clip we'll also provide, and then take a picture that includes that in the entire height of your tree. And from that, we'll be able to estimate the tree's height, and then with the diameter measurements, we'll also be able to estimate tree biomass. And so that'll also give us some important information. And so additionally, once we roll this project out and start getting people to participate, we're going to be asking a subset of participants to also participate a little bit further, and that would be by sending us some twigs so we can look for scale abundance on the trees across these different urbanization or latitudinal gradients. Also asking some folks to send in soil samples or leaf samples, ways we can determine tree, with some analyses we can do on those to assess tree condition and tree health. And eventually, if we do have enough participation, we hope to expand this to additional tree species. And so in summary, we want to monitor red maple growth with a simple instrument, a dendrometer. And this data collected will allow us to make better, have a better understanding of the relationships between environmental stress and pests, and then also between tree growth and these environmental services it provides. And also knowing about these tree conditions based on these based on warming or environmental stress, we can come up with better guidelines for putting the right tree in the right place and also create better estimates of carbon sequestration. And so you want to determine how urbanization, and so the reason we have this wide spread in the spread maple is we can have a better understanding of how urbanization and background climate affect tree growth and pest abundance. Because generally, unfortunately, warming from urbanization or from climate change can reduce tree growth. But as I said before, in some cases, it might make trees grow more and become healthier due to longer growing seasons. And so by having this wide range, it should help us get at some of those questions and then their answers. And effectively, when we know what trees grow well where or where they don't grow well, that should help us develop IPM and planting recommendations. And with these, we're hopefully developing educational materials for extension personnel, end users, as well as the public. So to change the planting practices in urban tree health, we really need to reach urban planners and professionals with some diverse extension products. And so here's again where you can go to participate. I'll put this in the chat box. Someone had asked previously. So hopefully you'll have some folks or yourselves that might want to help, and we'll get these being shipped out pretty soon. And again, we have a bunch of collaborators and cooperators, and thank you for your time.
Any questions?
Any questions?
Okay.
That's great.
All right. I wanted to thank Michael for coming to talk. We he and I had gotten together, and when he had brought up this project, which sounded like a great project, and so eventually this will be sent out, I guess, to the agents to inspire their volunteers to be able to participate in this project to document these red maples. And so, thank you. Anyway, so I just wanted to let you know that this may be coming your way, and as soon as it's available, I'll let you all know. And we can go ahead and move forward. So I'm thinking we are 9th year. Okay, so here we are.
Indeed, here we are. Hello, everyone. If you could give me a green check if you can hear my voice okay. I believe I clicked the talk button, and there we go. At least to see some people can. I want to make sure everyone is familiar with the workings of the interface so that we can do some interactive things here. The first of which is going to be a little poll. Thank you. Putting a check mark there. Okay, but what I want you to do is, this is Mike Munster, by the way, from the Plant Disease and Insect Clinic. My colleague Matt Bertone, entomologist, and I will be talking through the rest of this, except for the times when you will be doing some work that we're going to put you to. And here's the first one. We want to get a poll of people's years of experience being an Extension Master Gardener volunteer. And we realize that some of you are in rooms where there are multiple folks, and so you're going to need to put do some tallying. So among those with you, if you could add up how many people fall into each of these categories, the less than a year, the 1 to 5, 5 to 10, or greater than 10 years of experience as Master Gardener volunteers. And then what you're going to do is, a check mark is okay if it's just one, but what I would like you to do is use the text box, and the tool is located, one second, right about, on mine, it's right about there. There's a letter A with some lines across it, and you can use that, and then it will allow you to put a little box on the whiteboard and write the number in. So for example, I can do this. I have less than 1 year as an Extension Master Gardener volunteer because I'm not one, so I just put a 1 there. And see, that's how it works, which I will now attempt to take off the screen. There we go. Within that A, there are actually two different tools. There's one that will allow you to use just a simple text label, and another that gives you multiple lines for more text. I can see that people appear to be using both of those, which is fine. And getting an interesting distribution here. Quite a few folks who are newbies, which is great. That means Matt and I can repeat ourselves without feeling too guilty.
Yeah, that's our ulterior motive with this, I guess.
It also presents, obviously, a challenge to be able to address things that will fit the needs of both the newer folks and the more experienced folks. All right. See if I can do a quick screenshot here, and we'll take a look at that later. Okay. All right, moving along then. We would like everyone to take out a sheet of paper and number it from 1 to 5 because this is a pop quiz. This will be done individually. You will not put your answers either in the chat box or on the whiteboard. We want you to each write your own answers and not reveal them until everyone has had a chance, and then we'll go back through them. And the nature of this pop quiz is what I learned last year on PP&P. So I'll give everybody another 10 or 15 seconds to do that. Get out your favorite stylus, pen, pencil, quill, whatever you use. And let us begin. Question number 1. Which of the 3 rose diseases pictured here is only serious in the nursery, at least in North Carolina? In other words, there are 3 different diseases in this photograph, 2 of which are landscape problems and 1 of which is not a landscape problem but just a nursery problem. Which one is the one that you would not typically see in the landscape? In fact, I got it once as a sample from a landscape. It was because it had been recently transplanted from the nursery. So A, B, or C. Again, just write your answers on your paper rather than putting them into the system here. Question number 2. What's wrong with this tree trunk, if anything? A little bit hard to get a perspective there, but that is a tree trunk roughly at breast height. Question number 3. Which of the following is not a symptom of boxwood blight? Leaf spots, leaf drop, root rot, or stem streaks? Which one of those is not something that we associate with boxwood blight? And this is an important question. We've gone over this several times, but then I've been a little surprised that some of the information isn't really getting out there as far and wide as I'd hoped. Question number 4. Of these 4 images of tomato diseases, which one of them is late blight? This is from the plant pests and pathogens from last year. Almost the same exact slide. And question number 5, what are these? Should probably mention that that this is a branch here, so these are quite magnified. These structures are not 3.5 feet tall each, so they're not rain barrels stacked up next to each other. A couple of millimeters tall. Okay, so pencils down. Actually, we're not going to know if you have your pencils down or not. So do whatever you want with your pencils. And let's look at the answers here. Question number 1. Which of these rose diseases is only serious in the nursery? And that is downy mildew. For some reason, this time of year, cool, moist, so it hasn't been that moist. That is what really makes this thing take off. You can get a lot of defoliation, a lot of problems for your nursery growers, but it's not a landscape problem that you would be encountering from the folks that you're dealing with. Black spot, of course, on the other hand, by the dark spots and the feathered edges on them, yellowing and leaf drop as well, would be something that everyone sees on roses pretty much. And Cercospora leaf spot, a little bit different shape to the spot, but you can also get the yellowing defoliation. That's more of a hot weather pathogen.
All right.
Matt, take it away.
Many people know this anyway. This is a pretty heavy infestation of gloomy scales. One thing to note, well, first of all, the giveaway is that the trunk looks all pimply with little black circles all over it. Very rough appearance. And this is a very heavy infestation, but you get the idea. Another thing to note that I see in this picture is something interesting that people might not be aware of, but these little orange things on them are actually a parasitic fungus on the armored scales, a species of Nectria that we see often in these high populations. So sometimes people don't notice the scales as much, but then when these orange fruiting bodies come out, they notice that and they think, oh, is this an infection of the tree? Well, no, it's an infection of the actual scale insects.
Number 3. The answer is C, root rot. The fungus Calanetria pseudoniviculata causes problems on the leaves and stems of the boxwood, but does not infect the roots. So I like to say that look for these 3 things: defoliation or leaf drop, starting down toward the lower portion of the shrub and moving upward, dark brown leaf spots visible on both the upper and lower surface of the leaf, and the dark streaks on the green stems of the boxwood. If you see 2 out of 3 of those, you've probably got boxwood blight. There are some things that can fool you, but really keep those 3 things in mind as you look at people's boxwood problems. With the tomatoes, it's A. The late blight is the one with the grayish and more diffuse blotch on the leaf. It also can cause very extensive leaf death in a short period of time and fruit symptoms as well. If you're curious, B was a picture of tomato spotted wilt virus symptoms, more of a speckling on the leaf there, and C was septoria leaf spot, and D early blight. You can see the classic target appearance of the early blight lesions.
All right, so those were actually eggs. They weren't some kind of fungus or, or anything like that. We had a little quiz last year about some bug eggs, and this is a very common one that we see a lot, especially this time of year when there aren't any leaves and people are looking at their trees. These are the eggs of wheel bug. And this is our largest assassin bug. They are technically beneficial, but are generalist predators, so we'll feed on all different things. Soon enough, we should start seeing these young wheel bugs. This is a little later in star right here, but the very young ones are going to have a red abdomen and a black thorax and head. And they normally stay in a group near the eggs when they first start off, and then start to disperse as they get older. There are a few other assassin bugs that have very similarly oriented and batched eggs like this, where you basically have these columnar, kind of very tall eggs with a little cap, and they're kind of all held together by this sticky kind of resinous substance. That's very typical of wheel bugs and other assassin bugs, but not so much of some of the other types of bugs, as you saw last year, which will lay either groups of round eggs or barrel-shaped eggs that are shorter or different colored.
Say, Matt, have you ever gotten bitten by one of those?
No, and I plan not to ever get bit by one of them because it supposedly would hurt a lot. But I also try not to handle them. So, yeah. I have been bitten by a young green assassin bug, Zelis. It was just dropped from my ceiling in my home. They're common in homes around the winter time or fall. And dropped on my hand and bit me in the thumb. It was a little tiny one, but it still kind of hurt a little bit, and then it just itched. But if a tiny one can hurt, then I would not want to get bit by an inch-long assassin bug. So.
that makes a lot of us. All right, hopefully that was a helpful review. We're not going to ask for how you did, but we want to encourage people to, or just refresh people on what some of the points that we talked about last year were. And let us go on with the current and not so current diseases, and you'll see why I say the not so current in a minute. We are, of course, a week later than we usually would be. Instead of the 4th Tuesday of February, we're in the 1st Tuesday of March. And there's a saying in Mexico anyway, febrero loco, marzo otro poco, which is February's crazy and March a little bit more so. But I want to start out with a wonderful picture I took when we went with the family to Pullen Park here in Raleigh, a nice little park just adjacent to campus. If you haven't visited it yet, I would definitely recommend that. And as you walk along the north side of the small pond there, lake, there is a Spanish bayonet plant, which earns every bit of its name if you've ever encountered the tips of those leaves. But it had a striking leaf spot on it, even back in February when we were there, which I thought, oh, I'm going to collect this and talk about it for plants, pests, and pathogens as a current disease. And when, of course, take a closer look yet, Matt was kind enough to take these close-up pictures, and I thought, oh, great, we're going to have something like a Calototricum. This is going to be an anthracnose disease. And so I got all inspired to talk about the anthracnose diseases. So I'm going to go on a kind of a long digression, an ode to anthracnose. And we've mentioned some of these diseases over the years here in Plants, Pests, and Pathogens, but we've never collected them all together under one roof. And we're going to do that now. So the first thing is, if anyone knows what anthracnose means. We have a fresh $5 bill with Matt's picture on the front for the first person who can give a good definition of anthracnose. All right, I see there's someone asking if I could speak closer to the microphone, since the microphone's right on my ear here. What I can do is turn up the volume a little. How is that, Lynn? Does that come through better? All right, good. Let's see, does it reach his relative, his or her Aunt H. Rachnos? Close, Mark, but not quite. This actually turns out to be what I consider one of the hardest terms to define in introductory plant pathology, which I won't do yet. First, I want to just give a reference to where the term came from, which is awfully interesting. Went to one of my favorite references, which is the Oxford English Dictionary, and it turns out that anthracnose was actually a French word meaning charcoal disease, apparently coined back in 1853 or so to describe the disease of grape caused by the fungus Elsinoe ampollina. It turns out we do have this disease in North Carolina, although it is relatively rare, at least that we see it in the clinic. There's a lot of other more serious diseases on grape that we worry about, but it'll pop up occasionally. And for those who are really fans of etymology, the ancient Greek word anthrax denoted burning charcoal, hence the use of the word for things red in color, like a precious stone or a carbuncle, as well as for things black in color, like coal. So Matt and I were puzzling over that a few weeks ago, why this word would mean both things that are black and things that are red, and it turns out that because it originally meant burning charcoal. But that doesn't tell us what it means today as a disease of plants. So the best definition I could find, I hunted around a little bit, was from Schumann and D'Arcy's Essential Plant Pathology text, and they say it's a disease caused by a cervuli-forming fungi and characterized by dark sunken lesions and necrosis. So a couple of terms here that might not be handy. One is necrosis, which just means dead tissue. And the other is a cervuli, or a cervulus in the singular. And what that is a particular kind of fruiting body. This is my own graphic here, which is okay, but we're looking at the cross-section of, let's say, an affected leaf or a fruit, and in magnification, you get this busting out through the plant epidermis, and some of the fungal cells there lining the bottom of this saucer-shaped fruiting body and producing a bunch of asexual spores that we call conidia. And they are going to be in some kind of a matrix, a bit of a slime or ooze, and they'll kind of stick together there. And that is really important because these are diseases that are going to be spread by water splash, by handling plants when they're wet, but they're not going to be easily spread by just simple wind currents or air, as some other fungi are. So the interesting thing is the definition is a little bit overwhelming, but it does let us know two things. If you hear that something's an anthracnose, number one, you know that it's caused by a fungus. Number two, that there's going to be some kind of a dark sunken dead spot on the leaf, fruit, or stem. And number three, that it's probably going to be spread about by something like water splash or possibly handling, or I could see if I can think of an example right off the top of my head that insects might participate in this too. Here is an enlargement, so that white bar at the bottom is a 1 millimeter scale bar, showing you the cervuli of the fungus Calototricum breaking out through the surface of the plant, and the slightly salmon-colored mass of spores that are emerging from there. You may or may not, in the cervuli of these fungi, see what are called setae, which are the kind of pointed, usually dark filaments or spines that are coming out from here, the acervulus. And here you can see, a little bit difficult against the background, but there are the spores. And these large blocks here are the cells of that leaf. So that's what they look like microscopically. Let's see some examples here of some of the anthracnose diseases that we commonly encounter. If you see this, English ivy, if you see this symptom here in North Carolina, you immediately know you have one of two different things. This is either going to be bacterial leaf spot, caused by the bacterium Xanthomonas, or it's going to be anthracnose. And the way to tell the difference, well, for one thing, the bacterial leaf spot usually has a little more yellowing around the spot. And you could have both of them maybe in this picture. But the conclusive thing for anthracnose would be under a little bit of magnification, a hand lens, you'll see the fruiting bodies, those acervuli that have formed there. These will not be present, these little pimples will not be present if you've got the bacterial leaf spot. So in this case we know we have anthracnose caused by the fungus Calototricum trichelum, which is specific to Hedera, so English ivy, and I think it's Algerian ivy, its relative. Another area where you encounter anthracnose is in strawberry, although fortunately this is not something that the home gardener generally deals with. It's more of a plasticulture issue. But there are 3 different species of Calototricum that can cause anthracnose in strawberry, and there can be crown rots, there can be runner infections, or fruit infections. This is a pathogen that likes it hot and rainy, and fortunately it will not overwinter in the soil just by itself, but it will overwinter in crowns. And I've got a couple of, for more information on these two, I've got a couple of URLs which I will cut and paste here into the chat box. I see that Barbara has done one too. So Barbara, what was the URL that you posted there?
That was from a blog post we did some time ago about anthracnoses.
Oh, okay. Thank you. So question, still have your green arrows and red X's. Clear those out. Give a green check if you think that this is anthracnose, and a red X if you think it's not anthracnose causing this fruit rot on the strawberry. Looks like people are getting familiar with the tools here. That's good. Waiting for 15. See if we can hit 15. All right, the great majority here are in fact correct. This is not anthracnose, which you could tell because of the fuzziness. The fungus involved here, which is botrytis, is causing the lighter airborne spores to be produced on the surface of the fruit here. So this would not be anthracnose. Good. This one is anthracnose. It was actually a sample that came in from out of state, but there's the infection on the fruit. This can rot the whole fruit away if it's ripe. And you can just barely see some of the spores being produced on the surface of the fruit here. One that you will probably see, I believe I have in my own vegetable garden, is anthracnose on snap bean, caused by the fungus Calotropicum lindamucianum. So here the classic round sunken lesions. Sometimes they would be more of a brick red color. And the fact that it can be on the fruit, on the pod, but it also can be on the leaves and the petioles. But the fact that it can get on the pods is a bad sign, because it's something then you would suspect or be concerned that it could be seed-borne, which in fact it is. So that's when we get around to talking about control, will be one of our most important strategies. I'm not sure if it's the same species or another that gets on lima bean, but you notice this is a little bit different shape of the lesion, kind of a little more irregular, nice rusty color, but another anthracnose on the, this would be on the lima bean. A different one completely is the one that gets on the cucurbits. You can see the round, fairly round anyway, brown lesions that with experience you would not suspect this was downy mildew, although you might get confused at an early stage. If this were water soaked, you might confuse it for angular leaf spot. So this is an infection of cucumber leaves, and here's a picture of it on a watermelon stem down at the bottom here. This wouldn't be the first disease you would suspect if you saw a rotting stem on one of the cucurbits, but in this case that's what it turned out to be. The fungus is called Calotropicum orbiculari, and we would get it in most often on cucumber and watermelon, though other cucurbits are susceptible. And it's mainly something that's going to cause leaf and vine lesions, though fruit infection is not unheard of. So here's kind of the problem. Two sets of watermelon leaves that came in, set 1 and set 2. Which one has anthracnose and which one has something else? Well, we won't put you on the spot here and make you decide, but it turns out that number 1 is anthracnose and number 2 is gummy stem blight. So there is a big difference in those two diseases, but similar symptoms, at least on the leaves here. For example, there are resistant varieties for anthracnose, but not for gummy stem blight, I don't think. I'm not 100% sure on that. And so this is just an example of one reason why you're going to want a diagnosis, which is one of the list of recommended control and prevention methods. Now, after I put these together, I realized this looks like a generic set of recommendations for any kind of foliar fruit or flower problem in the garden, and so be it. The first thing, sow clean seed. So you would definitely not want to save seeds from an infected pod. You would, if you're a saver, if you buy seed, then make sure it's from a reputable source and that you're planting the ones that are good looking. Practice crop rotation. We preach that a lot. And destroy infected debris, because it will survive from year to year on that. So don't put those in the compost pile. Just make sure those go out into the landfill or the incinerator if you're a place where you're burning your debris. Minimizing leaf wetness. I already talked about how they spread around by water, but also water on the leaves is what gives that fungus the conditions that it needs for the spores to germinate and then to penetrate those leaves. So don't overhead irrigate. When you do irrigate, do it at a time, or you have to overhead do it at a time when they will dry quickly. Don't work among plants when they're still wet with dew or irrigation because you're going to be spreading things around. If you do see symptoms, get a diagnosis. Make sure you don't have something more serious than the anthracnose. My favorite story about that, a little bit of a digression here within a digression. When I first was working in Mexico, down in the state of Chiapas, I was in charge of a plant pathology laboratory at a center there, and my assistant had a sample of saw palmetto that had been brought into the lab by someone who was growing that down there, and had correctly identified black mildew on the foliage. But when it turns out when we got out to visit the field there, that was the least of their problems. They had that, but they had crown rot from Phytophthora. They had soils that really weren't the soils that plant should be growing on. So just kind of the importance of getting a diagnosis. And you may have anthracnose, may not be that big of a deal, maybe a moderately big deal, but if you've also got root knot nematode or Phytophthora or something like that, then you've got another level of problem. Now, the recommendation for control of anthracnose often includes switching to resistant varieties, but I put a question mark on that because I, scanning around a little bit, could not find in Ted Wiener's list any of the resistant varieties being the ones that you would find for home use. Now, there may be some out there, but they're probably more serious diseases that people are concerned about getting resistance to, so that the ones that are or that include anthracnose resistance are going to be in your commercial type varieties. Another whole set of anthracnose diseases are those that occur on trees. This one is on sycamore between anthracnose and bacterial leaf scorch. That's why so many of our sycamores look terrible around here. Very often with these, the veins are going to be affected, and the infection will look like it's following along the veins. And here's another one. So let me go. I do want to do this one, have your participation. So I'm going to change the polling to A through D. Now instead of the check and X, you'll have a choice of A, B, C, or D on the poll. So go ahead and answer this question. Is this an example of botrytis blight, dogwood anthracnose, septoria leaf spot, or spot anthracnose of dogwood? See how many people are signed on here. We got 45 people in the main room. So we had 15 or so the last time, which would be about a 1 out of 3 participation rate. See if we can do a little better than that maybe on this question. Right, I set a goal in my mind of 20. Hopefully we can see 20 people participating, answering. There we go. So let's go ahead and publish that response. So 1 out of 45 said B, 2 said C, 16 said D, and D is in fact the correct answer. This is spot anthracnose of dogwood. Now, how do we differentiate between these two? Let me just tell a tale of 2 anthracnose diseases on dogwood. The first one is spot anthracnose, which we will see in a few weeks here once the bracts start to enlarge. Depending on the weather conditions, we may just see some of the red spotting, or we may see very extreme deformation of the bracts, making it quite unsightly. In fact, there was one time when I think I was looking at this just from the road, driving by a home, but at first it looked like it was severe frost damage. So this can, in certain occasions, become very severe, but it's not a life-threatening disease to the tree. It just kind of ruined the aesthetics for that particular year. It does cause infections on leaves, and those do resemble septoria leaf spots. So the person who replied septoria was not completely off the mark there. And the rundown here, the summary is of course, symptoms being small reddish spots on the bracts and the leaves, which can become numerous enough to deform the bracts. The pathogen is the fungus Elsinoe corni, and it will overwinter in the twigs and even in the fruits that hang on to the tree to be able to start the infections new in the following year. It's favored by wet springs. Seems like weather really does determine how much we get of this disease from year to year. So the idea of using some kind of fungicides, that's a little bit dicey. There are programs that you can use, one that's based on 3 or 4 sprays of a protectant spread out across the year at key times. But since you don't know really how much you're going to have from one year to the next, you don't know if it's really going to be worth the time and effort and expense, and it may or may not be something that is going to be economically worthwhile. Again, this is a common disease, but it's not one that's serious. In fact, I think in Plant Specimen Pathogens in the, I don't remember what year it was, I asked folks for their crowdsourcing of whether they had seen it or not. So keep your eyes open. We may be asking about what diseases you've seen again. Now, I like to distinguish this between a disease that unfortunately has a very similar name, which is dogwood anthracnose, caused by the fungus Discula destructiva. And the symptoms here can begin with that leaf spot or blotch, but then the problem is that it'll move down into the twigs and into the trunk, causing even the death of the tree. This is worse in a forest setting than it is in the urban, more open landscapes. And in North Carolina, this is something that is restricted to the mountains. So it will be worst above about 1,800 feet in elevation. As you go farther north in the country, of course, the elevation doesn't have to be quite as high. And we haven't seen a sample of this come into the clinic for quite a while, but it is important to remember the difference between the spot anthracnose of dogwood and the dogwood anthracnose, which are quite different. So that was my digression into anthracnose diseases, all inspired by that Spanish bayonet plant, that yucca. And it turns out that it wasn't an anthracnose at all. After some incubation, we had a nice tuft of the spores of one of the cercospora-like fungi, and then some others that didn't produce those spores, which turned out to be a Leptosphaeria conitherium. I'm not sure which is the correct name. And so it was an inspiration, but it turned out not to be a good example for that at all. Going along with our current diseases, this is a sample that came in last month now of some Japanese holly, and I'd like to read you the description that was sent along with it. Hollies planted last spring at new ag building with shredded hardwood mulch. Fungal growth on mulch also found on plant stems, and in crown some plants are dying back dead. Also some planting issues, but curious if this could have caused any of the dieback. Does anybody know what this is? You can go ahead and write in the chat box or paste it to the whiteboard with the text tool if you'd like. No takers? All right, it is a little bit unfamiliar at this worn out stage of development. Maybe if I showed a picture of what this thing looks like at an earlier phase, you would recognize it. This good old Fuligoseptica, the dog vomit slime mold, starts out as that frothy, well, of course it starts out as a plasmodium. You don't even see it in the mulch, engulfing little bacteria, bits of organic matter, etc., causing really no harm, but suddenly decides it's time to sporulate, pulls together in a yellow frothy mass that then becomes the spore, just a mass of spores covered by a pink crust. So the picture that you saw earlier was the remnants of one from last year that had crawled up on that that holly stem. It had nothing to do with the decline of the plants. It was just incidental. Oddly enough, we got in the month of February something like, no, we got 3 samples of this disease last month, and that was compared to 2 samples of it in all of 2016, and I'm not sure the reason for that. But this particular disease on Pachysandra, which you can see here some of the symptoms. Let me read the description of what was sent in. It says, quote, "The home site is shady. Symptoms were first seen last summer. Pachysandra is in multiple contained beds, very large plantings of Pachysandra. Established plantings, 32-year-old plantings." Close quote. And so you can see the leaf spots here with some yellowing, the dead stem ends, and the defoliation. This is an extremely destructive disease of Pachysandra, and it's called Volutella blight. That's because the fungus used to be called Volutella, and now it is called, I'm going to take a deep breath, Coccinonectria pachysandricola. Now that's only 2 syllables short of supercalifragilisticexpialidocious. I think it's the longest scientific name that I have ever tossed out there on plant specimen pathogens. But you can just call it Volutella blight, and it is certainly a problem on Japanese Pachysandra. I am not sure if the reports of it occurring on our Allegheny spurge, the native species, are accurate or not. It is, though, very destructive on this particular host, and so here are some recommendations. Of course, make sure that if you're transplanting into a new bed or replacing plants in an old one, that you are using only healthy individuals that are not going to be carrying the disease with them. You don't want to plant this in full sun. It does want some shade. And you want to avoid stress, both drought stress on the one hand and overwatering on the other. Of course, injuries of any kind like salt from de-icing and so forth. Don't work in the bed when wet, similar to our anthracnose diseases, because you can spread the spores around. And avoid excessive leaf wetness by watering when the drying will be rapid, and thinning the bed or plants around it to promote good air circulation and rapid drying of the leaves when they do get wet from rain or irrigation. Remove the diseased material when you do start to see it, and again with a question mark as to whether fungicides would be worthwhile or not. We don't have a lot of fungicide information, but our NC Ag Chem manual lists mostly the protectants such as mancozeb, chlorothalonil, and the copper formulations. But you're going to have to be out there putting these on every 7 to 14 days depending on the disease pressure. So that's kind of a lot to ask to protect just a ground cover. And I think this is my last example on the current diseases. But first a question, still on the ABCD spectrum here, because the host is cherry laurel. And the question is, botanically speaking, cherry laurel is A, in the cherry family, B, in the laurel family, C, in neither of those families, or D, in both of those families. So please use your polling tool to choose one of those letters. Take about another 10 seconds. All right, looks like about the same number of people participating. And here's our breakdown. Move it out of the way. Okay, 3 folks said the cherry family, 15 said the laurel family, and 3 said in neither. A good thing no one said in both families. That would be an intrafamiliar hybrid, and I don't think those exist. But But it turns out that the three of you who said the cherry family are correct. It is a Prunus lorocerassus. And the disease in question here, sent in as an image sample by Catherine Holmes, Rockingham County, last month, is this. You can see the large holes in the leaves. And if anyone tells you that they know what is causing this, get out the extinguisher because their pants are on fire. It is really hard to say what this is. Now, we've got a little more luck with shot hole on something like an ornamental cherry tree, but I have never been comfortable in the saying exactly what is causing this particular disease when I've tried to isolate it or incubate it and so on. The important thing, of course, is that you understand it's not insect feeding. Of course, I'm talking about it, so that's probably your first clue, but also notice that there are spots, and I can show you a little bit of a close-up here that she sent in. There are spots where the infection has not yet dropped out. So you get this necrotic or dead leaf spot, and then eventually it drops out because the plant, Prunus species, have this wonderful ability to wall off the infection, and then it drops out. Now, there are a couple of fungi that could do this. There are there's a bacterium or two that could do this, and there are some other factors that may trigger this particular symptom. So it's not really easy to say what was causing it in the first place. According to some testing in Georgia about 20 years ago, it was the autoleucin among the cherry laurels that came out consistently the most susceptible, with the Zabeliana and the Schipkaensis causing or having sometimes, excuse me, less shot hole and sometimes not. But anyway, worldwide there's been, like I say, at least 3 fungi, a couple of bacteria, and some other things implicated, as well as, like we say, the suggestion that there's some kind of abiotic factor. Even if it is a disease, which it probably is, the infection could have occurred weeks, or in this case, months ago. So it's probably not the time, or unless it's getting around to that time again, when anything you would want to try and intervene would do any good. Again, really hard to diagnose, so sending in a sample may or may not be something we can give you an answer on. The important thing, though, is that as long as you're not seeing dieback, this is not something to worry about. If you are seeing the branches dying back, then we got to look and make sure you don't have a root rot or a peach tree borer or something like that going on. I would say that any kind of a recommendation as far as fungicides, even though the work in Georgia showed that there was some benefit to a fungicide like a mancozeb, I would say that for the homeowner, that's probably not ready for prime time there. This is just something that you want to explain what it is. Certainly make sure they don't go out and start spraying insecticides, but it's something that's going to be there. And I've got a good reference here on this, which I will also cut and paste to the chat box from the folks at Rutgers. I'm not sure I agree with them in their publication about the idea that winter injury could be a cause. So that does it for the current diseases, and let me just mention what to look for in the coming months till we meet again. On our woody ornamentals, of course, that's spot anthracnose of dogwood, which we talked about at length here. Camellia petal blight, the dying and falling off of the Camellia japonica flowers. And those of you who have been on before, remember you pull off the base of the bloom and see if you can see that white ring of fungal mycelium in the petals at the base of the circle of petals. That's what gives you the diagnosis. The gymnosperangum rust, we already saw one case, but it was from a greenhouse of quince rust on cedar. And of course, cedar apple rust coming out of the galls. Entomosporium leaf spot on Photinia and Indian hawthorn are pretty much there all the time, but you should be seeing those as people start getting out in their gardens again and wondering what's happening. And our root rots, of course, a continual problem in the landscape, both from Phytophthora and Armillaria, which are main 2 root rotting organisms in the woody ornamentals. On the veggie side, we are at the end of the crucifer season, but you still could see some sclerotinia stem rot or some black rot bacterial disease, Anthomonas, that's pictured on the right there, and some downy mildew is possible. peach leaf curl on fruits, and of course be working your preventive spray programs for your fruit trees. In the flower beds, black root rot of pansy and viola could be showing up, and botrytis blight as well as the winter flower starts getting at the end of its season. In turf, any turf species you might see a fair ring on. You could get yellow patch in your cool season grasses, especially tall fescue, and as the Bermuda grass and the zoysia resume growth, there'll be areas that are not coming back green, typical of our spring dead spot. And with that, I'll take any questions.
Mike, I have a question.
All right.
I'm
Yes.
having spinach that it did this in the fall and is doing it again, that just kind of melts down and rots right now. Is there anything that jumps to mind that would be attacking them? It just seems like the whole leaf gets soft and dies, like it's a root issue or something.
Did you say spinach?
Yes.
No, nothing jumps to mind. The thing that I would think of on spinach would be the white spot. I think the name of the disease is a Sarcospora or Sarcospora-like fungus, but that wouldn't cause a meltdown without first causing a leaf spot. So, yeah, that's something that we would need to see a sample of to figure out what was going on.
Okay. I might bring one in.
Matt, does that sound familiar to you at all? Anything that might be boring or swimming down on the base of that plant?
It's hard to say. There are some leaf mining fly maggots that mine spinach, but you would definitely, it doesn't cause a rot. You would see kind of translucent or bubbly kind of leaves with, and you'd be able to see a maggot inside if you were to break it open.
Okay, these are very tiny plants, so I don't know. I'll just grab a sample, see if I can get a good one.
Sounds good. Yeah, grab a couple of different stages so that we can try and catch whatever it is in the act. I see a question here from Jane that she had to leave and missed the presentation about anthracnose on dogwood. Yes, this has been recorded, is being recorded as we speak, and it will be available for viewing later online. You can get to the link for the recording from the PP&P website, and I'm not sure how long it takes before those are posted, but there's a process that has to occur before that happens. So Lisa can probably answer that question better than I.
Yeah, once they get it posted, I have to actually go in and request it, but then I will end up posting it on the Plants, Pests, and Pathogens. I think it's on the gardening portal. And so I'll go in there, and it will be posted there, and I'll put it on NCSU Garden as well. So you should be able to click it in a couple of different places, but if you want to do the MP4 version of it, it takes a while for them to process that. But as soon as they do, I'll have it up.
All right, so if there are no other questions about the disease side of thing.
Mike, there's a question about in the chat, best practices for black knot on plum trees, considering the variances in weather.
Oh, okay. I see it now. I did not see that when it slipped by. Also, someone who wanted to see the first Bolo slide. So let me go back.
Yeah, I put it back on there. Whoops.
Thanks. Black knot. Excuse me. I would refer you to, let me see if I can get it here quickly and post a link. There is a nice guide to disease and insect control in the home orchard. And while Matt is talking, I'll look that up and then pop that into the chat box for you so that you can follow that link. Thanks for catching that question, Matt. I missed it.
No problem. All right, if there are any other questions, if they're not, I will start talking a little bit about some insects. Well, we'll see about insects. All right. So today I'm going to just talk about one subject mainly, and talk about some bolos later on. But that subject is Columbola. Yay! This is a drawing done by a friend of mine, Thomas Shahan, who's an excellent photographer and artist. I, in fact, have a T-shirt now with this beautiful design on it. Columbola are a very important group of organisms that are encountered by people in various situations. You know, I've never covered them. We've covered them. I covered them actually a little bit when I talked about household organisms, household arthropods, and I'll touch on that a little bit later. But basically, Columbola are called springtails. So let's spring into spring with springtails. Columbola is an order of insects or hexapods, depending on who you talk to. Most recently, people have been considering Columbola and two other orders, the Protura and the Dipleura, mostly all soil hexapods, as non-insect hexapods. This is because they have a lot of different characteristics than typical insects that we would be seeing that most people are familiar with. Now, they are in the group of hexapods called the Entognatha. That literally means inside and jaws. And this is one of the characteristics that separate them from other insects. The Dipleura, Protura, and Columbola, the Columbola being the largest order in this group, with about 8,500 species worldwide, and certainly many more to be yet discovered and described, all have a mouth pouch with internal mouth parts that they can then push out and feed on the substrate. So rather than being able to see the mouth parts very easily, their mouth parts are internal. This is one of the characteristics of the group and what sets them apart from many other true insects. These are among the most abundant insects on Earth. I'll just call them insects for now. And there are estimates that there are about 40,000 individuals per square meter of soil. That means we did a calculation, a conservative calculation the other day, and there's several quintillion, quadrillion?
5 quintillion, I think it was.
Okay.
Or no, quadrillion.
Quadrillion, yeah.
In the state of North Carolina.
Yeah, so, I mean, it's definitely more than trillions of them. So they are just everywhere. Other than mites, they are basically the most abundant arthropod in soils. Now, nematode worms, being the animals, probably the most abundant animals in soils, but springtails are extremely common. Now, they are present in the soil and near the soil line, but they can also be found on trees, in puddles and bodies of water, and in homes. But the caveat is that they require moisture. They require high moisture because most of them breathe through their skin. Now, one of the groups that we'll talk about later on actually apparently has spiracles and tracheae, but most of them do breathe through their skins, which means they need to be moist to get that oxygen diffusion into the skin. Now, just showing some situations you might find them in. So other than the soil, there are often, and in fact I got a video from a friend of a relative wondering what these things were on a puddle. And basically on a puddle you may see all these floating little specks, kind of dull little bluish or blackish, or in this case when the person sent me, were kind of maroon looking. But these little organisms are springtails. Now here is shown Podura aquatica. This is the only species in the family Poduridae. And you can tell that it's a podurid rather than one of the other ones that you would find because it's got these long processes on the end, which I'll describe in a few minutes, which are part of what makes springtails spring. Now, there is another group of springtails, the Hypogastruridae, that will also be found on bodies of water, but that group is also very familiar in that they can be called snow fleas. in that they are sometimes found covering snow fields in the winter, and they're on top of the snow feeding on bacteria and other little microorganisms that are on the surface film. Now, on the snow, they look very obvious here. They're all hopping around. They're very tiny, like little pepper flakes. Most collembola are about 3 millimeters long or so. They range in size from 1/4 of a millimeter, which is extremely tiny, up to the largest springtails in the world are over 1 centimeter long, so almost 1/2 inch long, which is crazy. But most of them are going to be a few millimeters long. Now, the thing is that you can see these in the snow, but actually there are almost as many of these springtails around on the soil. It's just that people don't take a look in the soil. Now, I'm presenting on these today because, of course, if somebody's digging out in their garden, or they look under potted plants in their house, or other areas where there's a lot of organic matter, things like that, you may find springtails, and they are very abundant. So what do springtails do? So springtails typically feed on fungal hyphae, spores, microorganisms, and decaying materials. So you can find them in compost bins, but they're basically grazing small bits of organic matter and microorganisms from surfaces. And this photo down here by Andy Murray, I'm going to post a link to his Flickr page, because he's in Australia, but he's got some great photos that I'm going to use a few of in this talk. But this is a really cool little springtail feeding off of a slime mold fruiting body. So I've never seen a photo like this. I thought it was amazing. Now, again, like other Entonata, they have internal mouth parts. So they basically put their mouth up to the surface and stretch out their internal mouth parts to scrape the surface and feed on what is scraped off. Now, they do molt throughout their life. So, again, something different than many typical insects. Now, silverfish and some jumping bristletails, some of the more primitive of the true insects, will molt throughout their life. But springtails certainly do this as well. Another strange thing in comparison to many true insects is that instead of mating, having internal fertilization, what happens is the males actually lay down a sperm packet, which then the female takes up. This is actually similar to many arachnids, like pseudoscorpions and scorpions, and some other groups. Not spiders. Spiders do have internal fertilization. And actually, some of them, there's some really great video. I'm going to post now also a link to a video, have really cute, elaborate courtship dances. And you'll just have to go see the video to actually take my word for it. There you go. All right. So there's lots of different, with 8,500 species, there are dozens of families of springtails throughout the world, and many of them are actually very common. You just have to look for them, or if you're digging in the soil, you might see them. So I'm going to just go over some of the main groups that you'll see out there and discuss a little bit about their, what they do and how they look. Okay, first though, springtail anatomy. So springtails, this is showing one of the segmented springtails, not a globular one that I'll get to in a moment. But springtails are known for a few basic structures that they have. So the first, one of the most important one, is this thing called the caulifor right here. This is this little barrel-shaped organ in the middle, right behind their hind legs. This thing, nobody really knew what it did very much for a while until more recently, when they found out that it's actually a waterproofing organ. So even though it looks very small and stumpy, there's actually a long process that comes out of it, and they can actually wipe down their body with it to help waterproof them. Now, the spring part of a springtail, and why they're called a springtail, is that they have this long double or forked process called a furcula, and it's actually divided into a few parts which are important taxonomically. Of course, you'd only be able to see them under a very high-powered microscope because these insects are very small. But the furcula is here stretched out because the specimen is not alive. But in life, it would typically carry it under its body, and it would be held by these little hold points or little hooks called tenaculum, or a tenacula, one single tenaculum. And so in life, they would hold the furcula under the body, and they'd be walking around, and if something disturbs them, they release that furcula, and it launches them haphazardly into the air. They can't actually really direct where they're going to go, but they will flip around in the air and land somewhere, hopefully safe. Now, on their legs, the claws and these hairs and all that stuff can be very diagnostic for certain groups, but you rarely ever have to identify them. I've only had to ever identify them down to species a couple of times. Now, unlike true insects or non-insect hexapods, they don't have compound eyes but have numerous simple eyes, up to 8 on each side. And here's a really great picture again from Andy showing a very close-up of one of the non-segmented globular springtails, showing all those simple eyes on the sides. Now, they can lack eyes, as you'll see in a moment, or have up to these 8 ocelli on each side. So, going through some of the common ones you might find out there. So there's a general group we call the elongate springtails. That's because they're elongate. They're not the globular ones, and they're kind of not as pudgy as some of the other ones. And I'll go through a few families of elongate springtails. So the first is the Isotomidae. Now, a few of the elongate type springtails will basically have this very short neck. They'll have a little neck, but it's hidden. This front segment is hidden. And there really are no seeding on the top of that segment, and that's going to be characteristic for the next couple families. In isotomids, they have this little hidden segment here, but they also have fairly uniform length of their abdominal segments. So there's kind of the 4th abdominal segment, so which will be 1, 2, 3, 4. Is not longer than the 3rd, right here. So these are isotomids. They do have a little furculus. They can hop a little bit, and you can see the eye patches here. Entomobryidae is probably the most commonly seen family in homes and out in the environment of elongate springtails. Now, they do have the little neck where they have this hidden prothorax. You can see, really you can only see 2 thoracic segments, the rest of it's hidden. And they are similar to the next family and different from the isotomids in that this 4th segment, abdominal segment, is really large compared to the 3rd. So the 3rd is very small and this 4th is very large and long. You can see how large it is compared to the 3rd. Now in entomobryids, the antennae are the antennal segments are very similar. None of them are greatly elongate. And many of them also have lots of hairs, and a few have scales on them, just like butterflies and moths would have. Now, a very closely related family, in fact it was a subfamily of the Entomobryidae, are the Tomoceridae. Again, very similar looking. They've got a very tiny neck that you can't see in there, but there's a distinct constriction there. And again, they have a long 4th abdominal segment. Let's see, you can see it right here compared to the 3rd. And look very much like an entomobride, but the very good distinguishing characteristic is that this 3rd segment, antennal segment, if you see the 1, 2, and then this long 3rd segment, is extremely long and longer than the 4th if present. So in this one it doesn't seem to have a 4th segment present, but if it did, it would only be a very small segment. These also are sometimes found in homes, and of course in leaf litter and on algal. These rocks show a lot of algae that they're probably grazing. And many tomocerids have these scales all over the body. You can see a slight gray iridescence, usually bluish in tinge also, or purple. And again, very very flattened, modified scales. Another elongate springtail, which is somewhat closely related to those, but actually exhibits a different character, are the hypogastrurids. And these are the ones, the snow flea ones, and ones that are often on water. And they differ from the previous three groups in that you can actually see the pronotum, this prothorax, and it has hairs on it. So the presence of hairs on that prothorax can distinguish it from the entomobriids and the tomocerids. Now, in this group, they don't really jump because they either lack a furcula or have a very, very tiny furcula. Now, these, again, can be the snow fleas, and typically the two common genera shown here are usually, one is usually dark and one is usually lighter. But again, they're very difficult to identify specifically without looking at them on the microscope. Okay, so now some non-jumping springtails. I know the previous one was non-jumping, but it's technically classified with some of the elongate ones. These non-jumping springtails just kind of crawl around, and I think they're kind of cool looking. And there's a huge diversity of these. These two images look almost identical, except for the colors. It looks almost like a negative one of the other. But these are both neonurid springtails, and both are found on their bark, but they can be found in the soil as well, or crawling on surfaces with organic matter. So these are long, but don't have a neck. Basically, they have a head that's kind of connected and very, very gradually connected to the body, the rest of the body. They're also fairly pudgy and lumpy looking, so they've got all these little warts all over their body. Several of them are bright orange, like the one on the right, but I have found the gray ones and some other ones. These are the Neonuridae. Now, one of my favorites is the genus Moriolina. This is a photo I took out in Pisgah State Forest, and this is on a seep on some rocks that had a bunch of algae, and this little tiny springtail, only about 2 or 3 millimeters long, was crawling around in it, and it looks like a pincushion. Again, very warty appearance, no neck. You can actually see the ocelli here on the side of the face, and very short, stumpy antennae. So this is, again, another neonurid springtail. Another group that is very common in soil, these are among the smallest and usually are typically pale, but some are yellow. They do sometimes also congregate on puddles and other places, but if you basically dig into the soil and see these tiny little pale springtails, especially lacking eyes, these are the Onychiuridae. The furcula is absent in all these, and many of them also have these spines, so a pair of spines on the tip and the top of the abdomen. Now, the previous groups were all in one suborder, and they were called the Arthropleura, and Arthropleona, sorry. And those were divided up into segments, clear segments of the body. Now the globular springtails are a whole other group of springtails that, obviously from their name, are very globular and smooth. They look like a little tiny seed or something like that. Now, one of the most common families is the Dicertomidae. These are distinguished in the next group in that the last segment of the antenna, as seen with the arrow, is very tiny and pointed. It's almost like a little mitten in some cases because it may have a little point or thickening at the base. And these are extremely common. You often also see on the body, if you get up really close, you can see they have all these long crystalline-looking hairs or setae on the rump. I've found that if you, say, lay a blanket out in a damp lawn, especially in the morning before the dew has dried and the springtails are out and about, you can see them hopping around on the blanket or tarp. This was happened once at a yard sale we had when we had some stuff out on a tarp. We got lots of these little tiny springtails everywhere. Now these do have a furcula, a long one, and will jump. You can actually see the furcula on this one tucked underneath the body, and they will hop. Another common group, this is actually a few families that have similar characteristics. You'd have to actually look at the very specific characteristics to separate the families. But the Smithuriid-like, Smithuriid actually means field mouse, or Smithura means field mouse. So they thought the person who described these thought they looked like little mice. These are also globular and very similar looking to the dicertomids, except the tip of the antenna, if it's clear enough here, is divided up into many small sub-equal segments. But functionally, they're very similar to the dicertomids. There's another group of globular springtails in another family that are often found at marshy or wet habitats, especially along the edges of water. Now, that's a lot of different diversity. I hope you get something out of that as far as identifying some of the general types. All of these are fairly common. I've seen most of them in different situations. But if you want a little bit more information, I'm going to put up another link. This is to a really good website by a person who also identifies a lot of these springtails on Bug Guide. And Columbola.org, well, it's not really a link there. Anyway, if you search Columbola.org, lots of cool photos, lots of good information about the diversity of springtails. Now, springtails, good or bad? Well, the good is, and I think the good far, far outweighs the bad in springtails. The good is that they do help decompose organic matter and distribute microorganisms that also do so. So they help break down leaf litter and compost and things like that. They're also food for many organisms, especially very small other arthropods, predatory arthropods and such. There's something about, as far as respiration in kind of tropical and other soils, they may contribute to about 20% of the respiration of soils too. So they are kind of like earthworms. They can kind of help destroy organic matter and kind of aerate the soils, things like that. The bad, they may be a nuisance in homes. Now, some of the manuals say that they can bite, but they're really, again, with their mouth parts, things like that, there's no evidence that they bite. There was an old paper that had maybe some misconceptions about it, and it's been kind of perpetuated. They do not bite. People may be sensitized to their scales and hairs, I'm not exactly sure, or the dead bodies. And I have seen situations where hundreds of them can be dead in a bathroom, in a bathtub. Again, they're very well associated with moisture, so often you find them in kitchens, places where you have a lot of house plants or bathrooms. If you have an overabundance of them, it may actually indicate a moisture or mold issue in the home because, again, they rely on that moisture to survive and can feed on the molds and mildews and fungi. Controlling them, again, is going to be figuring out that moisture issue. Now, there is one species, Spinthuris viridis, by the name. It is one of the globular springtails, called the lucerne flea. And lucerne is just another name that British and Australians call alfalfa. And basically, this small springtail, it can be a major pest in some areas, especially in Australia and some Mediterranean climates. It will feed on the leaves of plants and cause holes and cause extreme destruction, especially because they can attain such high numbers in such a short period of time. And lastly, I just wanted to show some of the crazy springtails down here that Andy Murray took photos of. We actually have a very similar one to this here. This is a very humpback. This is actually an entomobriid, but it does have the iridescent scales like the tomocerids I showed. And this is actually close to a near-neurid, so kind of those little pudgy, warty ones. But instead of having just typical warts, this one has these very large orange projections all over the body. Okay, before I move on to the bolo, does anybody have any questions about springtails?
Matt, this is Mike.
All right. Mike, you got something?
Yes. Yeah. Why would they put waterproofing on their skin if they need it to be wet to breathe?
Yeah, it's a good. That's a good question. It's more so they don't get stuck in the liquids and things like that, because they're in this environment and they drown very easily. I think it does stay moist enough, and I think it's more like a. I'm not exactly sure, actually, honestly, to tell you the exact reasoning behind that, but it does help with their water regulation. It may also be waterproofing to keep the water inside the body, just like a cuticle would. And so, because they're so tiny, they dry out very quickly. And so, I'm not exactly sure. It may also just be so the sphintherids, the globular ones, are the ones that are known to have a tracheal system, which would then be more like a typical insect. So then it would be more, they want to keep moisture in and they can respirate through those tracheae. I'm not sure with the elongate springtails what the coliform is doing, especially for them. They used to think that the coliform either took up water itself, or coliform is actually, I think it translates to glue bearer, and that's because they thought it might also help them stay on the ground if they accidentally release their furcula, but again, there really isn't a lot of evidence for that. So very good question. I'm not exactly 100% sure on all the physiology of springtails, but it would be worth looking into a little bit more. All right, any other questions?
Matt,
yes.
I know they don't bite. I did have a client who was very irritated by them. So can their legs be something that would irritate, or any other parts of their bodies that would provide irritation to people that when they have the issue in their home, when they have moisture problems in their home and they've got them in their homes?
So, I mean, people can become sensitized to different types of the body parts of insects. I know, for example, that working with moths, people often get really sensitized to their scales and can become highly allergic to moth scales. It may be something like that. It may be something unrelated, that there just happens to be a lot of springtails around. And so it's... Again, I don't think it would be them crawling on you. They have very tiny claws. I would imagine that they are so small that they wouldn't be able to puncture or do anything to your skin directly. And also, they're really not commonly found on people. But it's hard to say whether it's something that's a contact allergy from dead bodies of these springtails, if it's something that's inhaled in the air that could cause that. And if they have a moisture issue, there could be other issues as well. So I don't think it's very clear exactly, but I would say there's really no way they bite, and they really don't come into contact with people very often. Okay. All right, some things to be on the lookout for. So insects are waking up, of course. If you go outside, you're going to see them waking up, but you sometimes don't have to go outside to see them waking up. We, in fact, got a couple photos on this top right here of these wasps that were waking up inside of an apartment. These are paper wasps, and typically paper wasps, but sometimes hornets and yellow jackets will overwinter in homes, and it's this time of year that they start to become active and want to go find a nest site. And so they will come out in the part of the home or in a commercial building. And we've had several of these cases over the years where paper wasps are emerging at this time. Also, stink bugs, some flies, things like that are going to be emerging. I've heard a few people complaining of stink bugs becoming more frequent in their homes. That is an issue. Now, it's usually the brown marmorated stink bug, and the other stink bugs can be there as well as box elder bugs, things like that. But mostly brown marmorated stink bug. Now, one of the ways to help reduce this, the numbers, is either by catching them by hand and putting them in some soapy water, or disposing of them some other way, vacuuming them if there's a lot, or I'll post a link right now. There are a few stink bug traps that you can actually make at your home that, especially when the house is dark, it uses a light to attract the stink bugs. They think they're going to be able to go outside. They get trapped in the trap, and you can collect many, many specimens by doing this. So again, be aware that if you have things in your home right now, they're just trying to find their way out. The best things to do then to discourage them next year are to make sure that the outside of the house is properly sealed so there's no routes of entry for these insects in the fall. But for now, you're just probably going to have to deal with them. Other things waking up are going to be the ground nesting bees. We've actually seen a few samples of these already. They're going to be typically found in bare soils, making lots of holes and pushing up lots of dirt. Just wait around a little bit and you should see the bees flying in and going into the holes. They are not aggressive. They're important early pollinators. And basically, they are a good sign of spring. And you will see them, large colonies of them. Now, a not so good sign sometimes, we are going to start seeing flights of termites, especially Reticulatories flavipes, the eastern subterranean termite. There will be another couple flights, one in June for Virginica and one in the fall for Hageni, different species of subterranean termites. Now, if you see these outdoors, or they're coming from a stump or somewhere in the wild, no worries. They're helping to decompose that wood, make it more into soil. If they're emerging inside or very close to a home, definitely get that checked out. They may be emerging from a colony that's in the wood in the home. Boxwood leaf miners, about the time it starts to warm up, check your boxwoods for swarming little orange flies. Here is a female laying an egg into the leaf of a boxwood. The males will emerge first and start swarming, and the females will emerge later, mate, and then begin to lay their eggs. And this is when they are most susceptible, the adults are most susceptible to chemical controls. Once the eggs are laid and the larvae are in the leaves, it's much more difficult to control them, and they really are only going to be present at this time of year, the adults. The rest of the year they're going to be larvae in the leaves and waiting till next year to exit the leaf, and you'll see little pupal skins coming out of the leaves as well, where the adult fly came out of. And lastly, who knows what kind of year this is going to be for the fall cankerworms. I really did not see many adults this year over the winter. We usually see on the buildings females either dropping eggs on the buildings, or males around lights. I think I saw one male and maybe a couple females. We had one sample come in of an image of a female in a home, but really I'm not sure whether that's just anecdotal, what I've seen, or whether we're going to have a good year for plants where we won't have many of the fall cankerworms. But time will tell.
But then won't it be a bad year for birds?
Yeah, maybe, I guess, or other insects. It's hard to say how much they depend on these specifically. But I would think that it would be so variable every year, and as we have seen over the past few years, that depending on these insects might be difficult. So if it is a big caterpillar year, maybe, yeah, the birds get a little fatter, have better, healthier children. But anyway, that's it. We're going to also be, I'll be mentioning some more bolo next month, or in 2 months. But basically, one other thing to look out for is scales producing crawlers. Once it starts to warm up, we may see some on the trees and on the plants, but that is also going to be an issue for April and May as well, and so I'll mention it then. Okay, are there any questions?
What about ticks, Matt? Are they going to start resuming activity here?
Yeah, I would think that once it starts to warm up again, you'll have some activity, and it's hard to predict. I don't exactly know when the first days of tick issues are going to be. It's going to be more frequent once it starts to warm up consistently, I think. I wouldn't worry on some of these days with very cold nights. I don't know that we'd find a lot out there, but as soon as it starts consistently being warm, I think we're going to have to look out for them. And you should always be careful and protect yourself when you're out in the woods, especially, or fields, things like that, because and do tick checks when you get home as well.
Right. If I may, going back to Sharon LaRouche, if I'm pronouncing your name correctly, Sharon, your question about the black knot, I'm sending out now the link to the Disease and Insect Management in the Home Orchard, which would be good reading for prior to next time's featured speaker, Sarah Villani, who's going to be talking about that very thing, about diseases in the home orchard. So follow that link, take a look at that. Don't forget to set your clocks ahead 1 hour before going to bed next Saturday so that you are not late for our next Plants, Pests, and Pathogens.
Okay, so don't go anywhere yet. We still have our showstopper plants. So that is coming up at the end. So should be next. And then about Sarah Villani, so that's going to actually be May 2nd. So it's the only other one that is going to be a different day, like our different start day this time. So we have one more thing to come, and I know we're kind of at the 12 o'clock mark, but we need to go ahead and do that. So everything finished with the bolos?
We are.
Okay. All right, let's see if we've got him in here. There we are. Okay, Mark, take it away.
Hey, so from those hexapodal harbingers of spring to a honey of a spring and summer berry, it's nearly time for strawberries, or at least it will be by our next Plants, Pests, and Pathogens talk. So even though growers were busy this weekend with frost protection, we can still look forward to these great fruits. Now,
I think I've messed up your slides. Can you want me to load them in?
Okay.
I'm so sorry.
Doing a great job. Lisa Sanderson, everybody. You little clap thing. Yeah, applause. Yay. Hey, why should we plant berries? Because they're delicious and because they can keep us healthy with their nutrition. So the deal is, across the United States, about 32% of our population, from the latest numbers, eat fruits twice a day, and barely a quarter of them eat vegetables 3 times a day. That's a total of 5 fruits and vegetable servings, and very few people are actually doing that. The numbers from North Carolina are right there on the slide. It's a quarter apiece. Good grief. How to plant? We'll do a soil test. Oh, but they're $4 a pop right now. What am I going to do? Hey, the value of the results are far exceeding of the investment of $4. You can barely buy a burger for that at a fast food restaurant, and you can eat all year from that soil test. So get that sucker done. Matted rows or hills? I'm so glad you asked. So we can plant a lot of our strawberries in the fall for a spring harvest, especially on hill production, but you can make a matted row right now. Do that as soon as you can get into the soil and till up a row and make that happen. So matted row production, you're going to get, so if you pull off all the flowers this year, you'll get great yields for the next 4 if you keep track of your diseases. So that's fun. When you're planting, don't go too deep. Don't go too shallow, Goldilocks. You got to get it just right. So what varieties? I'm so glad you asked. There's lots of NC State releases, including Eleanor Roosevelt's strawberries from 1939, or some more recent additions like Apollo and Atlas and Titan, Sumner and Bish. But the one that's listed on growing fruits and berries for the home garden is Early Bell, and that's an NC State release. How fun. I think I'm going to try Sweet Charlie this year. Now, Where can I learn more? I'm so glad you asked. Instead of typing in that whole long link, you can just search Grapes and Berries for the Garden, NCSU, from any search engine you choose, and it'll pop right up. I look for the purple one instead of the black and white knockoffs. Lastly, the professional folks are going to find a lot of great information, and you can too, right from our Strawberries portal. No matter how you like it, I prefer mine with the leaves cut off, making a little divot in there, filled with my own honey. But even if your strawberry shortcake isn't homemade, it's just as good as the cartoon. Thanks, everybody.
All right, so sorry, Mark. I have no idea how that happened. That was very bizarre. So everybody, he sent me really awesome slides, and they were very small, squeezed on top of other slides. Very strange. So anyway, I have a little bit of stuff to talk about, and so we've got we're down to the last chapter on the handbook. And so you can actually go to that website, go.ncsu.edu EG handbook, and you can see all of the chapters there, and Woody Ornamentals is coming. So you can check there on that. And then also the guidelines are here. So if you have questions about the Extension Master Gardener program, you can also get with me, but if there's things in the guidelines, I can have to help you with that. And here is Growing Small Farms, a great website. I think you guys have seen this before. I also want to let you know that you may be doing Costa Rica again in 2018. I just got back on Sunday, like at 1 o'clock in the morning, so not quite on schedule for me yet. But I do want to let you know that Extension Master Gardener College registration is supposed to start by April 1st, and I'm hoping a little bit before, and we'll have descriptions off to you about the session soon. So I'll have things on, if you go to the statewide page, there's information in the newsletter there, and so I'll have more information to you soon. So I want to say thank you for hanging on for another 6 minutes just to get this speedy information. And so thank you all for joining us today. And the next session will be on okay, I want to remind you guys that you can go ahead and sign out. They have not signed out yet. Okay, you guys still here?
I am.
Yeah. I am so sorry. I feel like I was completely off kilter today. And so I didn't know how to do the music, so thank you for doing the music.
Did you hear it? That was
I still couldn't hear it. Again, I get a prompt that says,
I heard it. I actually heard it
file?
twice at the end.
Yeah, I get a weird prompt that says, do you want to play this file? And I would it used to be that you just play it and I would hear it automatically. So I don't know what's going on with mine.
Yeah.
That doesn't matter. It's just me, then it's not a big deal at all.
Yeah, and I was hoping that Mark would stay in because I had never added slides before, and I think when I put it in, my little slides didn't show up anymore. It looked like it substituted his slides in, and it looks like it just copied his slides over the top. So that was really weird. So I feel guilty about his slides. But anyway, so I thank you all for being here today.
Oh, you're welcome. Welcome back.
Yeah. Well, thank you. And I got to the airport on Sunday night really late, and my car would not start. And it wouldn't start at all. Wouldn't do anything. So I ended up not getting home until really, really late. So
were you in
quite
Costa Rica?
back to normal yet. Yeah, it was awesome. It was a great trip, but it was really tiring. I think the flight part of it, just coming back, just long. But it was a really great trip. Yeah, thoroughly enjoyed it. And so I think. Sorry, go ahead.
Bit by ticks?
Oh, actually, yes. And you should know about that because I heard about him at the end. Yeah, Cindy and I were staying in the same place, and so I didn't turn on the heat, and she went and got a heater from somebody else, and so I had a heater, I just didn't turn it on. Thank God.
Yeah. Well, good luck. I mean, I don't think you. If you didn't get bit, I don't think they're much of an issue, too much, but it's better safe than sorry. But,
yeah, it did. And the weird thing is, it didn't look like a tick.
No, they don't, because they're a different family of ticks. They're the other
I'm going to
main
turn the
family.
recording off. Hold on.